Photosynthesis is important for plants because that's how they produce food. Plants gather energy from the sun and turn it into something they can use. Plants support all living organisms by making oxygen that we humans and organisms need. Photosynthesis occurs in the chloroplast of the plant and is divided into two different stages, the first being the light dependent and the other being an independent reaction. In the light dependent reaction, chlorophyll absorbs light from the sun. The energy that is gathered from the sun is stored as ATP and NADPH. This stored energy will facilitate the second phase. During this light-dependent phase, ATP and NADPH in combination with carbon dioxide is used to synthesize sugars. The second phase is called the dark phase. It's called this because it doesn't need the energy from the sun. It can also be called the Calvin cycle and it occurs in the stroma. The dark cycle uses energy that it's obtained from the previous cycle. All that happens in this stage is that the plant turns carbon dioxide into sugar. In this experiment, we were using spinach leaves to investigate how light would affect them. We used multiple lights to see the different types of effects. We had 5 different lights that we had to look at. Using 2 online sources, we observed how many bubbles were produced from having them under a regular yellow light. The experiment was done with different magnitudes of lights and the trials were held for 5 minutes. The plant was cut and placed into a beaker. The light was turned on and we observed how many bubbles were produced. The other lights were harder to observe because of the oxygen that was coming out. Blue light was producing the most oxygen, red light was producing a good amount of oxygen, but it's not as much as the blue.
Materials that were used for this experiment were spinach leaves that have been humidified, 3 petri dishes per team, bicarbonate solution, 100 milliliter beaker, cutout plastic bowls, and for those make it 3 as well. And the procedures that we use for this experiment starts with step 1. So there will be 5 different light conditions that you will be choosing from. Step 2, take several spinach leaves that have been humidified in the water. Step 3, place them onto one another. Step 4, using the small spectrophotometer tube, cut the disc. Make sure that you cut enough to fill half the tube and do not apply too much pressure because that could break the tube. Step 5, then you will take the disc out of the tubes then combine it with disc from another tube, into another different beaker that has sodium bicarbonate in it. This will extract the air from the disc. Step 6, connect a vacuum device to your sink, then connect the beaker to the vacuum device. Then open the water faucet all the way. You should start seeing air bubbles on the top of the disc. After 15 seconds of using the vacuum, stop, open flask, and then mix the container and repeat again 2 more times. You should now have a beaker with bicarbonate solution and spinach disc at the bottom. Step 8. Now in a petri dish, pour a little of the liquid from the beaker to fill it up. From the bottom of the beaker, take 10 to 15 discs and put them in the petri dish. Repeat this step 2 more times as well. Next, you will be working with the light conditions. Using the light conditions, cover each petri dish with a different color bowl from the light condition so that the discs receive different light, causing the effects of photosynthesis.
So, what good is an experiment without any documentation? So now that we've gone over some background information and materials and procedures, we can now go forward with our results. So now that we have our different petri dishes, we can now cover them with our bowls. We have 5 different colored bowls, which are either going to be green, red, blue, dark or black, and white. The reason for the different colored bowls is to observe how color and wavelength affect photosynthesis. As photosynthesis proceeds, the oxygen produced will refill the intramolecular spacers in the spinach discs and the discs will float again. So the hypothesis that we made before the experiment concluded that red or blue would work the best, green would have no significant impact, and dark would work the worst, and white would act as a control. So one of the reasons that we concluded that green would be the worst color is that plants already have chlorophyll, and chlorophyll is a green pigment that gives plants its green color. So since the spinach is already green, we decided that it would be harder for green light to be absorbed since the plant is already green. The reason that we said red or blue could be better candidates is that green is in the middle of the color spectrum, and red and blue are on the opposite sides, so it's possible that either color could be better for absorption. We said dark would be one of the worst colors because that means the spinach discs would receive little to no light, which is essentially like growing a plant in the dark. We said white would be the control because there is no radiant color for the spinach leaves to absorb, meaning that the plant would act in normal standing conditions like if it was being grown outside. And sure enough, our predictions were pretty spot on with our results. After concluding the percentages of discs floating, we were able to see that blue and red were the highest increase in percentages, with blue being just a little bit higher than red, and there was a big decrease in the dark light, and there was little to no increase or decrease with green or white, but there was a slightly higher increase in white. Our experiment shows that light is required for photosynthesis to work, as proven by the dark light, which had a huge decrease in percentages of discs floating. Our experiment also showed that color can increase or decrease the rate of photosynthesis, as shown by the increased percentages by blue and red. So even though our predictions were pretty close with the results, it was still fun to see whether blue or red was going to win and be the best absorbed color. And with that being said, let's move on to the conclusion.
Our hypothesis was right. The plant under the red light grew the most because it has longer wavelength. The red light source made the plant grow faster and taller because it had the right amount of energy and it was able to absorb the correct amount of wavelengths and light. Chlorophyll does a very good job of absorbing light under the red and blue. However, it does a very poorly job absorbing light from the green electromagnetic spectrum, which causes the chlorophyll to be green. Therefore, the green light is not absorbed, however, it is reflected, which makes the plant appear as green, and the chlorophyll is found in the chloroplast of the plant. Chlorophyll type A absorbs the most pigment in plants. When it's absorbing light, the wavelengths that they're able to absorb go up to 430 nanometers when it's blue and 662 nanometers when it's red, and then again, the green light just wrongly reflects, and so it appears green to us.